Great. Okay, I just want to introduce myself a little bit more and give a sense of what I work on and why fMRI methods matter to me in my research. So at a high level, our lab is interested in understanding one of the most classic and long-studied problems in vision science, which is when pattern light hits the retina and is transduced into neural signals, trying to understand how those signals are transformed along the cortex into a useful and meaningful representation of what's out there in the world. And what are the biological organizing mechanisms and constraints on learning that help us understand how to recognize people, recognize objects, move around, and make visually guided actions? So I try to chip away at this big question in my research, and here are some examples of projects I've been working on over the past few years. So first, along the ventral stream, we have these category selective regions preferring faces, scenes, bodies, and words. So I'm curious to understand why we have regions for these domains but not for others, and asking, are these regions better thought of as kind of independent modules, or perhaps differently as interrelated subsets of a representational space that's capable of telling all objects apart? So definitely some very contentious theoretical debates there, and that's one project I've been working on. Another one has been a collaboration that has recently revealed the existence of certain areas of visual cortex that have highly selective responses to food images. And so this is now adding on to our knowledge about category selectivity in the visual system. And this discovery was made possible by having new access to large-scale, really rich visual fMRI datasets that sample responses to many different kinds of images, to much broader set of stimuli than previous studies. So this is a second one, and another collaborative project I've recently worked on takes kind of a hyper data-driven approach to understanding the visual representations, taking around 200 neural network models, which are being used more and more in the field, and mapping them onto visual cortex and trying to understand whether there's anything systematic about what makes certain models more predictive of data from the visual system than other models. Maybe it's the training data set, maybe it's the architecture, maybe it's the task. So these different projects are all kind of different ways of getting a lens on these fundamental mechanisms and constraints that govern how information is organized in the visual system and how we represent different kinds of object categories. And there's a common thread between them in my research, which is that they all depend on having these high quality, condition-rich fMRI data sets. And the strength of the insight that we can derive from these kinds of experiments really depends on the quality of our data, whether we've been able to accurately measure brain responses in our subjects, and kind of reliably estimate how much each part of the visual system responds to each stimulus. So it's really essential that we have good methods for processing our data sets, and that's where GLM Single comes in. It's exactly the goal of the technique, and that's what I'll be talking about for the rest of our time here. And we can leave, again, plenty of time for questions or discussion. If people are curious about those other projects, we can also chat about them at the end. So just brief overview, I'll first talk about why we went about even developing GLM Single in the first place, provide some detail on how the technique works, and then cover a few implementation guidelines that'll hopefully help you all implement GLM Single on your own data sets if you're interested. And I do have a demo. I don't know if we'll have time to dive into it today, but at least I'll put the demo out there and people can follow the URL, which basically shows kind of a walkthrough of how to apply the technique. And yes, leave time for questions. All right, so why are we spending our time on this? The field of neuroscience is in the midst of a major paradigm shift toward data-hungry approaches. And so, as mentioned, we've seen this rapid increase in the scale and the depth of our data sets, where traditional fMRI studies maybe had a few hours of scanning per subject at most, but now we find ourselves in this regime where we have data sets like NSD, the natural scenes data set, perhaps some of you have heard about it or used it before, sampling tens of thousands of different stimuli at high resolution. And as I alluded to, you could use this kind of data set to answer many different kinds of questions. You could study representations in the visual system or in other parts of cortex. You could build encoding models that try to predict brain responses. You could study individual differences between subjects. Really just a whole wide range of things are possible. But in all cases, there's a fundamental limitation, of course, which is noise. And in fMRI, noise is the primary obstacle that limits the robustness and replicability of our studies. So I want to be more precise about how I'm defining the concept of noise. So here I'm assuming that we have fMRI data, and our goal is to fit a general linear model, or GLM, to estimate brain responses to each condition or each trial in the data set. And I will just provide, I'm sure this is review for many of you, but we'll provide an overview of fMRI, of GLM analysis in a moment. But for the time being, imagine we have a toy fMRI study where for each voxel in the brain, we're going to try to estimate a GLM beta value that reflects the degree of activation. And so in this toy example, we can pretend we have three conditions, which are inspired by some of my hobbies. So we'll say that we're showing people basketball, a picture of a piano, and a picture of a poker chip. And each one, of course, has a theoretical ground truth activity level in our voxel. But of course, we can't really know this ground truth. If we had a noiseless, parallel universe, maybe we could measure it. But our reality is, of course, very noisy. And so our measurements will look kind of like this. We might be able to repeat the experiment over and over and get kind of more pink dots. But just in general, what I'm referring to when I say noise in fMRI is this variability in our response estimates over repetitions of the same condition in our study. And of course, there are many different sources that contribute to this noise, which make statistical estimation very hard. Head motion, physiology, scanner noise, etc. And so this is a challenge just in our GLM procedure, and it becomes even more challenging when we try to pack our pack our experiment full of many conditions, where we run into a trade-off where increasing the number of conditions means necessarily we have to present each one fewer times, and that makes it harder to average together the pink dots to get an accurate estimate of the ground truth. So given all these different factors, the question is, how can we combat noise to harness the potential of existing and future fMRI datasets? And that's exactly the goal of GLM Single. So we've tried to come up with a flexible and data-driven, and the data-driven part is critical, flexible data-driven approach to denoising and signal estimation. And the key insight of the approach is to acknowledge that noise structure differs between subjects, between regions, and between even different voxels. And so our strategy should be to try to efficiently maximize data quality at the level of individual voxels and individual subjects. And so then whatever analysis we're trying to do, whether we're trying to study how brains relate to one another, or how brains relate to models, or how brains relate to behavior, hopefully improving the quality of those signal estimates will have a positive impact on our downstream analysis outcomes. So now, just again, a high-level overview of GLM analysis as it happens under the hood. Given some fMRI data, our goal is to describe the time series from each voxel, which is like the Y, as a linear combination of regressors that are contained in our design matrix X, with the coefficients, or the betas, reflecting the activation magnitude of the voxel. And so I'll walk through the process of how a design matrix would be constructed for one run of our toy experiment involving these three conditions. So the starting point is going to be a matrix that defines the onset times of each condition in the run. So let's say in this case, we have 5 instances of each condition, so 15 trials in this run. And this is what our initial matrix looks like. And now there are different ways that we could model these data, but let's, for this example, say we wanted to do what we'd call single trial response estimation, where we're going for 1 beta per trial, not 1 per condition, but 1 per trial. So we're getting a separate estimate for every single time that basketball is shown. That means our design matrix is going to start with 15 columns, 1 for each trial ordered chronologically. So now we've gone from a matrix of conditions by time to a matrix of trials by time. But we don't, of course, expect our brain responses to be 0s and 1s. So to finish building this design matrix, we can then define a hemodynamic response function, or HRF, which is the expected response profile of our voxel when it's stimulated. And then we, in constructing the design matrix, convolve the HRF with each column in order to get something like this. And so this is what we'll actually be putting into our GLM. And then by solving the linear equation that maps these predictors onto the observed neural time series, we can estimate a beta weight for each trial in the experiment. And then we can store those betas for further analysis. Those betas will be the inputs to whatever kinds of experiments we want to do after we've finished preprocessing our data. And as mentioned, the data are going to be super noisy. And so for this reason, it's really a common thing in the field to include nuisance regressors or noise regressors, and to kind of add these as additional predictors in our model. So for example, you might throw in some polynomials to model low frequency fluctuations in the time series that could be due to subject motion. Let's say they very gradually move their head over the course of the run, ever so slightly. We might want to try to model that using a polynomial. So now the design matrix will have a couple more columns, and if we assign some weight to those also, hopefully we can clean up or denoise our signal estimates. And this is just a generic framework for single trial GLM. And so I'll be referring back to the schematic as I talk about different ways that GLM Single tries to improve on this basic framework. Right off the bat, I'll say the only thing GLM Single needs as input are time series data and the matrix of the condition onset times, the first thing we discussed on this slide. And everything else covered here happens under the hood. And even though the tool was designed with this sort of single trial event-related experiment in mind, it's important to say that this is compatible with block experiments as well. In fact, that's something we've tested out in our GitHub repo. We have an example script just showing how this would look if it were applied to a localizer experiment where stimuli are presented consecutively within blocks. And it would also potentially work with a continuous fMRI design, like a movie or listening to a radio talk show, as long as these condition onsets reflect some kind of discrete event happening in the experiment. For example, a character coming on screen. So I can pause here. I've just laid a lot out there. Before I talk about how each step of GLM Single tries to improve the quality of these betas, I can pause if anything was unclear so far, if anyone has questions.
I have a question, Jacob. Can you hear me? Okay, cool.
Yeah.
Do you assume the same hemodynamic response function for every voxel, and do you have those parameters for that fixed?
That's a great question. In fact, that is the target of the first major optimization within GLM Single is to try to come up with a optimal custom HRF for each voxel. So it is possible to use GLM Single if you wanted to assume an HRF that's the same everywhere. That's simply changing a flag. You just have to say, I want to assume the same HRF, and it'll work. But the default is to choose one for each voxel, and in a few moments, I'll go into exactly how that works.
Okay, cool. Thanks.
It's already unmuted. It's already unmuted. Quick question. The HRF convolution, which do you guys use? Is it just like a simple Gaussian, or is it like double gamma or whatever it is?
I missed a part of your question, but
which HRF convolution do you use?
I think it's just the it's the standard approach that is typically done in GLMs. Kendrick, I don't know if you want to jump in to say which function it is specifically.
Sure, I can jump in. If you're using it in kind of a canonical sense, like assume one that fits all, it's a double gamma that was derived from some empirical data. But as Jacob's going to explain, the library approach is a bit different, although the form of the library is also roughly double gamma. In fact, we fit the double gammas to each of the empirically derived libraries, and it fits quite well.
Great, cool, thanks.
Thanks, Kendrick. Okay, moving on. Let's get into talking about how each of the optimizations happens within GLM Single. High-level overview, the tool is basically implemented in a single function, which is available both in MATLAB and Python. And as I said, it takes as input this design matrix, where each column indicates the onset time for each condition and the fMRI time series data. And this can work in either volumetric or surface space. And it returns as output an estimate of single trial bold response amplitudes. So under the hood, there are these three methods that help optimize the quality of our beta estimates. The first is the use of this library of hemodynamic response functions, which we alluded to a moment ago, that seek to identify the best fitting HRF for each voxel. And the second is a method called GLM denoise, which basically involves coming up with data-derived nuisance regressors that are used to remove noise from the beta estimates. Hopefully they'll be more kind of targeted to the specific characteristics of a subject's noise. Third is an implementation of ridge regression as a method for dampening the noise inflation that can often happen when our GLM predictors are close together in time and highly correlated with one another. And I'll go more into that in a few slides. So each procedure here tackles a different component of the signal estimation pipeline. And then when we apply them in conjunction, we finally get this estimate that is a beta coefficient reflecting percent signal change in each voxel in response to each condition. And as I mentioned earlier, it's possible to pick and choose if you did want to use one, but not all of these or two. There are ways to easily turn them on and off, and there are other hyperparameters that users can adjust in order to tailor the pipeline to their specific needs. So before I jump into actually talking about the methods, I did want to quickly just motivate, because I know it can get pretty dense, to kind of re-motivate, like what's the downstream impact? Why do we care? Why might we want to use this? And just briefly highlight the major take-homes from our recent paper, which came out in eLife, describing what happens when you apply GLM Single. And what we did in the paper was we basically took two of the biggest data sets we could find, natural scenes data set and Bold 5000, and we performed a series of comparisons that basically measure the impact of GLM Single on the quality of our signal estimates and on different downstream analyses of interest, just comparing between a baseline GLM, which would be kind of the typical approach, and GLM Single, so that it'll be blue versus red in these graphs. And the first thing we noticed was that measuring the reliability of our response estimates, we can see that GLM Single on average tends to improve the reliability in terms of like, reliability in this case defined as consistency of the estimates over repeated presentations of the same images. And GLM Single improves over the baseline. We also found that GLM Single tends to decrease dramatically the amount of temporal autocorrelation in our betas. In this case, temporal autocorrelation refers to how similar the spatial beta maps are between consecutive trials. And this is just visualized over the course of 3 runs here. It's actually kind of striking how much of an issue that was using the generic kind of GLM approach. We also found that GLM Single tends to enhance the representational similarity structure that we observe between different subjects, and that we were able to decode individual kind of one versus all decoding of single stimuli improved dramatically when using GLM Single, especially in the most reliable voxels. So altogether, we were pretty surprised actually at how much of a difference these kinds of techniques made. Okay, now let's get into how each component of GLM Single works. Oh, and just to provide a graphical intuition for what it means when we have these more consistent GLM estimates at the level of single conditions, this was an analysis we actually went beyond NSD and Bull 5000 to look at a third data set, which was publicly available from the Study Forest repository. And this is a auditory task where subjects listen to 25 different genres, or 5 different genres of music in the scanner, 25 different total conditions. And on the left side, you can see what happens when we use the baseline GLM. Each column is a condition, each dot is a trial. And so when we compute split half reliability, you can see it's above 0, but it's not that high, and there's quite a bit of spread within each condition. And then applying GLM Single, the effect is pretty striking, and you can see a lot more of clustering around, hopefully, the ground truth, presumably a more accurate estimate of the response in this voxel to each of our conditions. And this is a cherry-picked voxel, to be clear, but as we show in the paper, this is a widespread effect. Okay, so jumping into each component of the pipeline. The typical approach in fMRI, of course, is to assume that the response profile of a voxel follows the shape of the canonical HRF, which was originally derived from studies of primary visual cortex. It's actually a pretty strong assumption that this should be the same HRF throughout the brain. And I think more and more people in the field are talking about different ways of relaxing that assumption. So the consequence of having the wrong HRF can be pretty severe. And in fact, mismodeling it can really increase the amount of noise we have in our single estimates. So GLM Single relaxes this assumption. And the HRF selection strategy that we employ is pretty simple, where we have this library of 20 candidate functions, and essentially we try to select an optimal HRF for each voxel, one from the set of 20 in the library. And under the hood, what happens is the data are basically fit once using each of the candidates, and because it's a relatively constrained library of only 20 options, it's not going to take that long. It's actually a pretty efficient procedure to identify the optimal HRF. For each voxel, we just choose the one that has the highest variance explained in the time series. And again, the user has full flexibility to enable or disable this procedure, or even to add additional HRFs to the library if they were interested in doing so. So I can pause here, and I know there might be other questions about HRFs, so people should jump in if anything is unclear. Okay, great. So that's the first component. Now on to GLM denoise. So as we've said, fMRI data typically suffer from large amounts of spatially correlated noise, for example, due to head motion. And so to account for this, again, a typical setup is to include these nuisance regressors, try to account for different components of the time series that are not originating from cortical or basically non-neuronal sources of noise. And again, we're often trying to model the effects of motion, skull, white matter, etc. But GLM Single takes a different approach, which is basically to try to directly model components of the data that are likely to be noise. And so the method is to derive a set of optimal noise regressors using GLM denoise, which was developed close to a decade ago by Kendrick. And at a high level, the procedure works as follows. The first step is to fit an on-off GLM. And so this is a GLM where instead of trying to model responses across the different conditions, we're simply collapsing all of the conditions onto a single predictor and just trying to find parts of the brain that are active in response to the experiment. And so once we've fit that on-off GLM, we can separate out the group of voxels that seem to care about the experiment and those that don't. And so the voxels that are not active we'll call the noise pool. They just don't seem to respond to experimental stimuli. So then we can take our data from the noise pool only, and the next step is to apply PCA. And so we take those data, we try to understand the structure of the responses in the noise pool, and then we use cross-validation to iteratively include one by one the top principal components from the noise pool as candidate nuisance regressors in our model. And so the goal of the cross-validation procedure is to figure out what's the optimal number to include to maximize the variance explained in our model. And again, hopefully cross-validation will help you not include too many or too few. Of course, the consequences of including too many could be that you start removing components of interest or somehow biasing or overfitting. And so polynomial regressors are also included automatically, which is important to mention. And when you do this GLM denoise procedure, you'll come up with interesting looking, potentially structured. It's kind of hard to tell. It's hard to interpret what these PCs actually represent, but this is the kind of thing you'll come up with. And as a standalone technique, GLM denoise has been shown to outperform a bunch of other denoising techniques in terms of the cross-validation accuracy of GLM estimates. And more recently has also been shown to boost the replicability of cross-subject RDMs in representational similarity analysis, and also to improve decoding, both within and between subjects. So this is the second major component. I'm happy to pause here if anything was unclear.
Just a quick question. So, I mean, so you talk about comparing the old approach, which can enter, sort of regress out things that you expect to be interfering with your, what you want to actually look at. Whereas this new approach sort of takes the brain activity itself and tries to find those brain activities themselves that it thinks are not involved in the task and then regress those out. But with your toolbox, could you still add something externally that you know is causing noise, such as say the button presses they're doing like sort of motion, motor movements, etc.
Yeah, that's a great question. Yeah, the toolbox is flexible in that users can certainly include other noise regressors if they wanted to. So it's not required by default, but as with the HRFs, there's the ability to, if you had a priori a reason to believe that a certain component of the signal should be regressed out, you could also include that on top of GLM denoise.
Awesome, thank you.
Okay. So now onto the third component. So imagining in our toy setup, we've identified our optimal HRFs. We have our nuisance regressors from GLM denoise, and now comes the task of actually generating those final estimates of activity in each voxel. And of course, because the time series of an HRF is very slow, taking around 30 seconds, fMRI designs, which often rely on ordinary least squares estimation, often suffer. And so I'll tell you how that unfolds. The issue is that there's substantial overlap in the responses across successive trials, right? And so when our regression predictors overlap, this is just a fact of ordinary least squares estimation, it's hard to come up with accurate betas, again, due to this multicollinearity. So thinking about this concretely in terms of our design matrix, let's just imagine we have these two predictors, and you can see they're pretty close together in time, and they have these long time courses, so they'll be highly correlated. We can just imagine that it's r equals 0.8 in this case. And so let's see what happens just in a toy example if we wanted to estimate these betas. So the ground truth, we can imagine is there, and there's this phenomenon where if your predictors are highly correlated, what will inevitably happen is that as one beta goes up, the other inevitably goes down. And so you could imagine running this experiment over and over, and you might estimate the first black dot in your first experiment. You run the experiment again, you get the second black dot, and you do it over and over, and you'll generally have this anti-correlated structure between your two betas due to the correlation between your predictors. And this is unstable. Right, it's just hard to know how to appropriately divide weight between your predictors. And so to solve this problem, GLM Single is going to apply a efficient reparameterization of ridge regression, which is kind of the canonical technique used to deal with having correlated predictors in regression models. So this is aimed at addressing the problem of having noisy estimates in ordinary least squares. And so the goal of the regularization is to shrink the betas in a manner that slightly biases them toward the ground truth. And so, presumably, you could imagine shrinking your betas, but it's not necessarily the case that any mode of shrinkage will, draw them closer to the ground truth. So the goal of ridge regression is, again, you could think of each of these green paths as representing some amount of regularization. And so if you were to regularize all the way, you'd of course end up at 0. And you can just think of what ridge regression is doing under the hood as biasing the estimates in a manner that will hopefully bring them closer to the green dot. And to do this, you do need to come up with an optimal degree of regularization strength, and that is achieved in fractional ridge regression using cross-validation. So you could even simulate this kind of scenario just to prove that it does what you want it to do. And I'll just try to describe what that simulation looks like. So you could imagine, again, having two betas, and you could simulate 200 different experiments. So each experiment is a black dot. And again, each of these trajectories represents a different amount of regularization applied using fractional ridge regression from 0 to 1. So if you were fully regularizing and fully shrinking, you of course end up at the origin. And what frac ridge achieves is the optimal amount of regularization per voxel in order to bring the estimates closer to the green dot. Right, and compared to the ordinary least square solution, which tends to add noise. And the specific advantage of frac ridge or fractional ridge regression as it's implemented is its efficiency. And so it has an optimized mode of sampling different regularization levels in order to quickly achieve an ideal estimate of how much the regularization should be. And it's outperforms other methods that involve just grid searching over different log space lambdas. So this is a bit confusing, but I'm happy to also just take questions if anything is unclear. Okay. So at a high level, that's the overview of GLM Single and what's happening under the hood. 3 different methods for addressing different components of the signal estimation pipeline. Again, it's can be a lot to wrap your head around. So if anyone has questions, please jump in. At this time, we can talk about some practical guidelines to help people implement this if they're interested. One of the first questions that we get since releasing the toolbox has to do with experimental design. So which study designs are compatible? And there are a few things to consider. It should work in a straightforward way with event-related and block designs. Importantly, GLM Single is not appropriate for using with resting state data because the data lack any kind of event structure. Again, the pipeline depends on the existence of some kind of discrete experimental condition. So if you have continuous designs like movie watching or listening to stories, as I said earlier, that could work as long as you code different events as discrete repeated instances. Or if you had like a language experiment or an auditory experiment, you could code different words or different parts of speech even as events. In terms of how to prepare the time series data, again, it works with either volumetric or surface data. And in terms of pre-processing approaches that are compatible with the pipeline, motion correction is fine and slice time correction is fine. Those are both compatible. It's important to say that there's no need to filter the data, high pass, low pass filtering ahead of introducing it to GLM Single, just because the pipeline is aimed at kind of understanding the structure and achieving kind of a more tailored method of cleaning up the data than would be achieved by simple filtering. Spatial smoothing and registration methods are compatible. As mentioned, GLM Single does by default expect there to be some number of repetitions in the experiment. And this is important for specifically those cross-validation procedures in GLM denoise and FRAC-ridge. And again, in GLM denoise, the goal was identify how many noise principal components to include in the model. And for FRAC-ridge, the goal was to understand what's the optimal amount of regularization to apply at each voxel. The more repetitions you have, the better you'll be able to estimate those hyperparameters, because the way that cross-validation happens is basically predicting repeated data from separate runs than the ones that are being processed. So if you have a greater quantity of repetitions, you'll be able to more accurately do that leave one out prediction. That said, it's not necessarily required that you have tons and tons of repetitions. And the proof is kind of in the pudding. For example, in NSD, there are only 3 repetitions of each condition, and the technique seems to work pretty well there. So more repetitions are ideal, but not required. And then in terms of how GLM Single fits with existing pipelines, like SPM, FSL, AFNI, what have you, the key thing is that this could really replace the existing GLM components within those pipelines. And so, especially for experiments that might not play super well with existing GLM frameworks, if you have tons and tons of conditions, or this is really meant to be kind of an easy to apply generic framework that could hopefully without too much effort work with most data sets. And again, if you only want certain components of the pipeline turned on or off, that's easy to achieve via hyperparameters. We've made a couple resources available for users. There's the GitHub repo, and this is at www.glmsingle.org. Redirects here where you can find the code and other documentation, including several example scripts. And these show basically step-by-step process of applying GLM Single to data from a bunch of different contexts, event-related data, block data. kind of provides a helpful walkthrough for users, showing the process of setting up a design matrix, setting up your data, inputting it to the function, modifying different hyperparameters, etc. And also then interpreting the outputs and showing kind of some basic quality checks and sanity checks that you can do. And of course, there's the paper, which provides kind of a more in-depth coverage of the different statistical issues that are at play here, and kind of a deep step-by-step look at the method. If anyone's curious to try the demo, now or later, it is at tinyurl.com/GLMsingle demo. It's a Google Colab notebook. So basically just click File, save a copy in Drive, and then you can modify it, edit it, run it, however you'd like. And this Colab demo actually uses an example data set that's taken from the natural scenes data set. So it's a small subset, and it should only take a few minutes to run, but hopefully we'll provide a window into, provide a framework or a template for application to your own data. That's really all I had prepared to talk about. So if anyone has questions, happy to keep the discussion going. But yeah, that's a high-level overview of GLM Single. And again, just want to thank my wonderful collaborators in this project.
All right, thank you so much. Thank you, Jacob. Feel free to come up if you have any questions. Here.
Hi, Jacob. Very nice talk, and I really like your really like this method. I just have a quick question. Since you have like 3 components here, and each of them you need to select the best like parameters or something. So when I choose like all 3 on, how much more time I need to spend for the computer like to get the best solution compared to the baseline model you give here?
Mm-hmm. Yeah, that's a really good question about runtime. I think it depends on several different factors. First and foremost, how much data are you trying to process? So if you have, a small scale data set, you could, I don't know what small scale means in the context of the kinds of experiments you're interested in, but assuming that you're not trying to process, gigabytes and gigabytes of data all at once, the pipeline is very efficient. And it's usually, just to provide a benchmark, I can process, one session of data from the natural scenes data set, which is pretty hefty in terms of the number of trials. I think there are 750 trials per session. Should only take a few hours to process using all the optimizations that are included, assuming you have enough CPU memory. If memory is an issue, it's possible to handle that by processing just smaller groups of voxels at a time. So one of the hyperparameter options in the pipeline is called chunk num, which basically defines the number of voxels that you want to pass in at once. So one thing that affects runtime, of course, is how much data you're trying to process. And also you can, if you have a lot of memory, you can process more data at once, and that'll speed it up if you're able to kind of take bigger chunks. If efficiency were a concern, another thing you could do is kind of put a cap on how wide of a sweep you want these different cross-validated components to explore. For example, in GLM noise, let's say that the default, in fact, this is true, the default number of noise PCs that are tested, in other words, like by default, the cross-validation procedure is going to explore anywhere from 1 to, I think, 12 PCs, and so fit a separate model for each level and stop when cross-validated variance explained is maximized. if you didn't want to go all the way up to a ceiling of 12, or if you said, I'm concerned that this cross-validation procedure will take too long, you could set a lower cap and say you only want to explore, up to 3, for example. Similarly, in ridge regression, the default is to explore evenly spaced regularization fractions from 0 to 1, and I think they're spaced by like 0.05. But if you wanted to cut down on the runtime and sample fewer, of course you could do spaced by 0.1 or spaced by 0.2. So at a high level, I think the answer is it depends on the amount of data you have, depends on the amount of compute you have. But even if you're constrained, there are ways to modify the pipeline to make it more efficient if it really is an issue. But we've been analyzing some of the biggest data sets out there, and it hasn't caused that much of an issue so far.
Okay, thank you.
Any questions in person or on Zoom?
I have one. Can you hear me? Good?
Yeah.
Okay. So I can see how this makes a lot of sense with the single trial estimates, and you say it works for block design too. Is there a big advantage of using it for block design over like a just standard like FSL pipeline? Have you looked into that?
Yeah, that's a good question. We haven't directly compared GLM Single against FSL, AFNI, all the other options that exist. But I think the advantage of GLM Single with respect to block designs is kind of similar to the advantage of GLM Single with respect to event-related or any other kind of experiment, which is that you can take advantage of these data-driven optimizations that might not be available in other contexts. So, for just at a minimal example would just be like HRF fitting. I'm not super familiar with FSL, so I don't know what options are available in FSL to customize HRFs, but we've found in our analyses of block data here that it actually does seem to make a big difference, even if that were the only thing you were going to, the only optimization you were going to include from GLM Single. Analyzing, for example, localizer data and still allowing HRFs to vary across cortex or across the brain, that itself seems to confer some benefit in terms of the accuracy of your estimates. So I'm not sure, Kendrick, feel free to chime in. My sense is that the optimizations here are not necessarily contingent on or tied to event-related data.
Yeah, I'll just briefly chime in. Yeah, in terms of comparing to other packages, I mean, the packages vary a lot. It's a lot of the nitty-gritty details. It's very hard to say anything general. One actually has to sit down and do it and think about what each implementational details what each package is doing. So we don't really have anything concrete necessarily to say there. Regarding the block design, it is, I think, true. I think common wisdom is, with the block design, differences in HRFs get less and less important. Like if you're essentially convolving a double gamma looking thing with like a 20-second long square wave, little differences in HRF are largely going to be small by the time you do that. But that isn't to say that there's still potentially benefits.
Thanks.
Well, I have one sort of kind of broad question. So you mentioned in the sort of implementation guidelines that the more repetitions, the better for this toolbox, for GLM Single. And I'm wondering, just sort of broadly, what you see has the greatest potential for this GLM Single, like for in terms of like getting more reliability in those, say, small data sets or small ends, or perhaps with higher level of precision of the strength of the scanner for smaller voxels. Where do you see this helping the most in terms of like what type of data set you may have?
Yeah, that's a good question. I think it can help in different ways for different contexts. I mean, the pipeline was designed kind of with an eye to these massive, large scale data sets, in a sense that it's almost required if you're going to have so many conditions and potentially few repetitions of each, to achieve any semblance of accurate estimates. It seems really necessary to think carefully about the assumptions you're making, and to have some amount of flexibility built in to your estimation procedure in order to be able to tell apart fine-grained differences between conditions. So I do think one of the biggest potential areas for impact is large-scale, naturalistic, high condition number data sets, but certainly not limited to that. And I would say that we haven't tested GLM Single as extensively on, for example, cognitive or more behaviorally relevant data sets that might look at kind of decision-making processes or other aspects of kind of higher level cognition, but we do feel, that there is potential benefit there. And so I think there are potentially applications all across the field of neuroimaging, whether or not you're interested in specifically like perception. There could be clinical applications. There could be, again, those higher level cognitive applications. And the reason is that these optimizations are not, they're kind of generic in the sense that there are potentially ways to improve upon kind of canonical modeling procedures, whatever kind of data you are interested in collecting. Just to give an example, we have a collaborator who studies language and has a very interesting data set where each condition is only repeated once. And so kind of generically looking at the implementation guidelines for GLM Single, you might think, okay, well, can I even use this tool here? Is this technique even relevant because we don't have these repetitions, and therefore how can we estimate these different hyperparameters? But we're finding that it's actually possible to derive benefit even in that context through a couple clever manipulations of the technique. And just one example would be like taking different subjects, and if you have one repetition per subject, even looking at the structure and the repeats that exist between people could be enough to come up with kind of a rough estimate of what those hyperparameters should be. So all of that just involves a little bit of like understanding the technique and then applying it in a way that's appropriate for the data you have. I just give this as an example to illustrate that I really do think it's potentially broadly applicable, and kind of we're learning more and more about the different ways that people are able to apply it, even if you don't have a data set that's kind of like NSD or that is just a standard kind of vision task. Kendrick, feel free to add on.
Yeah, I'll just chime in. It's a good question, a very broad question. I'll just comment on a few things that come to mind. I think the general mindset of block versus event, I mean, it's kind of hokey in the end, but it's kind of heuristically valuable because if the question is what is this good for? With block designs, they're typically for experimental situations where you have a few conditions of interest, like 3 or 4, and in some sense you design your block to elicit a lot of neural activity appropriate for those conditions. And in those types of regimes, you're basically overpowered, and like the data from a given subject is probably as good as quality as you can hope, and you have pretty good estimates of all the various beta weights or response magnitudes. So in those cases, you probably don't care necessarily about SNR in your estimates in those types of paradigms, versus the other end of the continuum, which is more like these event or condition-rich data sets where you have very brief neural events and very few repetitions. So I think that's a useful heuristic. Another thing to comment on, it actually gets more to kind of signal processing and what's the nature of these statistical methods. I think you alluded to kind of like different ways to acquire fMRI data. So if you're pushing the limits, like going to high field or trying to go high resolution, either in space or time, then the nature of the fMRI data is definitely going to change. And the way it will change will interact, in a sense, with these components that you're staring at on the screen. So just for example, thermal noise will increase as you increase spatial resolution, and you can think about each of these components one by one and think about, is that actually relevant for thermal noise? GLM denoise, these nuisance regressors, for example, aren't really, because these are the structured noise components that are kind of spatially correlated. So the GLM denoise component isn't really going to help you there. However, the first and third ones here are definitely relevant for increasing amounts of thermal noise. Anyway, it's a longer discussion, but yeah, I would say if you were thinking about, especially if you were thinking about atypical or pushing the envelope on acquisition styles, then these various components in GLM Single might be especially important to consider.
Well, I'm sure there are many here who could find this much useful for a lot of our neuroimaging studies. So I want to thank you, Jacob, one last time, and also Kendrick for chiming in as well. Really appreciate this discussion.